Hello and welcome. This video is for AQA A-level biology. The topic is biological molecules. We'll cover everything you need to know for that unit. Timestamps in the description below if you want to jump to specific topics. If you find this video helpful, please give it a like and don't forget to subscribe for more A-level content. Let's get started. We'll start off by looking at monomers, polymers and carbohydrates. Monomers and polymers. Monomers are small units which join to make large molecules, for example polymers. Here are some monomer units. These join together to make larger molecules, for example polymers. Here's our polymer. And our polymer can break down into the monomers. Polymers are molecules made of a large number of monomers. If you're asked to define polymer in the exam, that's what's going to get you the mark. Examples of monomers. Monosaccharides, these are monomers that make larger carbohydrates. Amino acids, these are monomers that make polypeptides, which then make proteins. Nucleotides, these are monomers that make nucleic acids, for example, DNA. We need to know and remember those three. Condensation and hydrolysis reactions. Monomers are joined or separated by chemical reactions. Here's two monomers. They can be joined by a chemical reaction, and in this case, it's with the loss of a molecule of water. This is a condensation reaction. There's our water molecule. Here's two monomers that have already been joined by a chemical reaction. We can actually use a water molecule to split them apart. That's a chemical reaction as well. That is a hydrolysis reaction. So, a condensation reaction. This joins two molecules with the formation of a chemical bond and the loss of a water molecule. Hydrolysis reaction. This breaks the chemical bond between two molecules with the use of a water molecule. Let's take a closer look at carbohydrates. Large carbohydrates are made from monomers called monosaccharides. We just talked about that. An important monosaccharide is glucose. This is glucose. The formula is C6H12O6. You probably remember that from GCSE. And here is a simple version or a simple diagram of a molecule of glucose. There is, of course, a bit more detail. And this is the detailed structure of a glucose molecule. We can't see the carbon atoms in this diagram, but we should note that they are in these positions here. And we can actually number these carbons. This is carbon 1, carbon 2, 3, 4, 5, and at the top there, carbon 6. However, we don't need to know the diagram in this level of detail. The specification draws it in this level of detail. Please note the position of the carbons. They are still there. The 6 carbons that we talked about, numbered as before. Next, we need to mention isomers. These have the same formula, but a different arrangement of atoms. So if we draw out our glucose molecule here, and another one next to it, we can see the atoms are arranged slightly differently. If you look at the OH and the H groups on the left-hand side one, they are actually flipped around on the right-hand side one. The one on the left is called alpha glucose, and the other one is beta glucose. You should recognize those two and be able to identify them based on the position of the atoms as shown there. So these two are isomers of glucose. Two monosaccharides can join to make a disaccharide. Examples of disaccharides are maltose, sucrose, and lactose. Many monosaccharides can join to make polysaccharides. Examples of polysaccharides are starch, glycogen, and cellulose, and we need to know about those for this specification. Monosaccharides and disaccharides. Disaccharides are formed by joining two monosaccharides via a condensation reaction. For example, here is a molecule of alpha glucose. Next to it is another molecule of alpha glucose, and they can join by a condensation reaction. This is done by the loss of a water molecule like so. There's our loss of the water molecule and the formation of a bond. A condensation reaction produces a glycosidic bond. So there is our disaccharide, which has been bonded by a glycosidic bond. This is our glycosidic bond here, and the unit is called now a disaccharide, a double sugar. This one is actually maltose. Let's put that other diagram back so you can have that for your notes. Examples of monosaccharides and disaccharides. Monosaccharides are glucose, fructose, and galactose. Examples of disaccharides that we need to know about: maltose, sucrose, and lactose. We should know the names of these monosaccharides and disaccharides and what the disaccharides are made of. So maltose is 2 glucose molecules, sucrose is glucose and fructose, and lactose is glucose and galactose. Worth remembering that all the glucose are alpha glucose isomers. Polysaccharides. Polysaccharides are formed by condensation of many glucose monosaccharides. First example is starch. We mentioned this before, and this is one that we need to know about. Starch is a storage carbohydrate in plants. Starch actually is a form of storing glucose. And starch is a polysaccharide formed by the condensation of many alpha glucose molecules. If we were to look at a cell, we'd see some starch grains. There they are in a dark blue color. So how is it formed? So here are a few alpha glucose molecules, and as before we have condensation reactions that join these together. So the condensation reactions happen as shown, and when they join we have a larger molecule, a polymer, that looks like this. We can simplify it to look like this, and we'll see why we do that in a moment. So we simplify, and there's 2 ways in which those chains can be arranged. One is in a kind of coiled or spiral shape, the other one has branches. Starch is a good storage substance. So here are the 2 starch structures. The one on the left is called amylose, the one on the right is a amylopectin. Those 2 are not mentioned on the AQA spec, however you can be credited for mentioning them as part of the structure of starch. Starch is coiled or branched in its structure. That makes it compact. A lot of glucose can be stored in a small amount of space. It's insoluble. That means it does not affect water potential. In other words, it does not affect osmosis. Water does not move into or out of cells as a result of the presence of starch. Starch is sometimes branched, for example in the amylopectin. This is so glucose is quickly and easily released for respiration. Glucose is released from these ends there, and because there's more ends, more glucose can be released for respiration. Starch is a large molecule, and that means it cannot cross the cell membrane, so it cannot leave cells. Very useful for a storage substance. Let's take a look at cellulose. Cellulose makes cell walls in plants. Here is the cell wall of a plant cell. It's formed by the condensation of many beta glucose units. Here is a beta glucose molecule, and several are joined together, but it's worth noting that alternate beta glucose molecules are inverted, or in other words flipped by 180 degrees. So if you look at the second one, that's flipped, and the fourth one, that's flipped as well. Every other one is flipped by 180 degrees. So we can see our condensation reactions happening like so. So we end up with a structure a bit like this. We could simplify it to look like this. This is a straight chain. So cellulose is made of long, straight, unbranched chains of beta glucose molecules. The chains are joined by hydrogen bonds to make something called microfibrils. So we have a bunch of chains together, and these are joined by hydrogen bonds. Hydrogen bonds are weak, but there are many of them to give cellulose its strength. Here's our microfibril. I've only drawn 3 chains there, but there actually is around about 30 or 40 chains that make a microfibril. Many microfibrils join to make fibers, and the many hydrogen bonds between the chains give strength to cellulose. Let's take a look at glycogen. This is a storage carbohydrate in animals, found mainly in muscles and the liver. It's a polymer formed by the condensation of many alpha glucose units. So here are the molecules in glycogen, already shown with their glycosidic bonds, done just as it was done for the starch. Glycogen molecules have many more branches than starch molecules. So here is an example of our glycogen, much more branched, and there it is drawn again, and we can look at the structure versus the function of glycogen molecules. It's coiled and branched. That means it's compact. A lot of glucose can be stored in a small amount of space. It's insoluble, so again, it does not affect water potential, does not affect osmosis. Glycogen is highly branched, so more ends, so more glucose is easily released by hydrolysis for respiration. You can see there's many more ends here than there were in our starch molecules. And finally, glycogen is a large molecule, which again means it cannot leave cells, good for storage. Let's have a little summary of the 3 polysaccharides that we looked at. So what are the monomers for starch, cellulose and glycogen? Well, for starch is alpha glucose, cellulose is beta glucose, and for glycogen again it's alpha glucose. What's the function of each? So for starch, this is a storage molecule, cellulose is for strength and support, and glycogen is for storage. What's the monomer arrangement in starch? It's coiled or branched. Cellulose, we have straight chains, and glycogen is highly branched. Starch is found in plants and algae, cellulose in plants and algae as well, and glycogen is found in animals. So that's the summary of the 3 polysaccharides that we need to know and remember. Next, we're going to take a look at Benedict's test for reducing sugars. All monosaccharides are reducing sugars, and some disaccharides, for example lactose, are also reducing sugars. This is how we do the test. Here is our reducing sugar solution, and here we have some Benedict's reagent. We pour the equal volume of Benedict's reagent into our sample, and then we heat our sample in a water bath. We would eventually see a color change, and that color change indicates the presence of reducing sugar. So let's take a look at the method. 1, we add an equal volume of Benedict's reagent to our sample. 2, we heat in a water bath. And 3, the formation of a red precipitate indicates the presence of a reducing sugar. Important to note, the color gives you an idea of the concentration of the reducing sugar in the sample. Here are a range of colors that the sample goes through when it's being heated with Benedict's, and the least concentrated is the green color. It goes through a red, an orange, and eventually a brick red precipitate color, and that's the most concentrated. You could probably see for our sample above it's nearer the concentrated end, but it's not the most concentrated. Remember, a precipitate is an insoluble solid. The mass of the precipitate formed indicates the concentration of the reducing sugar. So in other words, the mass of our brick red precipitate or our orange precipitate will also give you an indication of the concentration. So we filter after we've done our test, we dry the precipitate and we weigh it, and the mass will give us an indication of the concentration of the reducing sugar in the sample. For non-reducing sugars, the solution remains blue with the above test. There is no color change. What we can do is do the Benedict's test for non-reducing sugars. Here is an example of a non-reducing sugar. This is sucrose, which is a disaccharide. We can go through a hydrolysis reaction. In other words, we can hydrolyze the sucrose and break it down into glucose and fructose, which are both monosaccharides. Therefore, they are both reducing sugars. So this is how we do the test. Here is our non-reducing sugar solution sample, and here is some dilute hydrochloric acid. We add the acid to our solution and we heat in a water bath. This is the first step to hydrolyze the non-reducing sugar. Once we've done that, the solution is going to be acidic, so we neutralize and then we add our Benedict's reagent. We would then heat as normal as we did before and look out for our color change. So here are the steps. 1. Add an equal volume of dilute hydrochloric acid and heat in a boiling water bath. This will hydrolyze the non-reducing sugar. We would then neutralize with sodium bicarbonate and test to make sure it is no longer acidic. For this test, we would use either pH paper or a pH meter. We wouldn't use universal indicator because that would interfere with the color. Next, we then add an equal volume of Benedict's reagent and heat in a water bath. The formation of a red precipitate indicates the presence of a reducing sugar. Worth noting, however, if we are testing an unknown sample for a non-reducing sugar, we would do the reducing sugar test first to ensure there is no reducing sugar present, then we would do the non-reducing sugar test. We have now the iodine test for starch. What we do is we add iodine solution, which is orange, to our sample. A color change from orange to a blue-black color indicates that there is starch present, or starch is present. Here we have a spotting tile with 4 solutions in there. Here is our iodine solution. We can see there is starch present in the first one, starch present in the second, no starch present in the third, and in the final one we have starch, but there is a lower starch content, so the blue-black color is not as intense as it was for the other 2 above. Next, we're going to take a look at lipids and then proteins. Lipids. These are fats and oils, and they are found in both plants and animals. If we were to take a look at some lipid, in this case it's an oil from a plant, we would see that there are molecules of what we call triglycerides. These are triglycerides that make up lipids. Let's take a look at one in more detail. We have the glycerol at one end, and we have 3 fatty acids. This is the structure of a triglyceride. If we were to look at one of the fatty acids in more detail, we could see that we have part of it which we call the R group, that's the long zigzag chain part of it, and we have another part that we call the carboxyl group. Together, these make up the fatty acid. Let's take a more detailed look at the glycerol part of our triglyceride. Here's the glycerol, and this is the structure of the glycerol part of our triglyceride. Here's a slightly more detailed view of our fatty acid. We have a carboxyl group at one end. This one shows a bit more detail in terms of the arrangement of the atoms. And of course we have the R group. And as we saw, there are 3 fatty acids that can join a glycerol to make a triglyceride. So the way it works is we have a condensation reaction with the removal of a molecule of water. And then this will lead to our bond, which is called an ester bond. There's the ester bond there, and there are 3 ester bonds, 1 for each of the fatty acids. This is the structure of our triglyceride. Let's take a look at the R group in a bit more detail. The R group is a hydrocarbon. The R group can be saturated or unsaturated. If we were to take a look at the R groups in a bit more detail, the R groups are hydrocarbons. You might see something like this. The top hydrocarbon is a saturated hydrocarbon, meaning there are no carbon-to-carbon double bonds. The second 2, these are both unsaturated hydrocarbons. That means there is 1 or more double bonds between carbon atoms present, and we can see those here. So those 2 are unsaturated hydrocarbons with the presence of carbon-carbon double bonds. So, in summary, triglycerides, we have glycerol and 3 fatty acids. The fatty acids are described as having this general structure, COOH-R. The top one here you can see is a saturated fatty acid because there are no double bonds, and these 2 are unsaturated fatty acids. We also have the presence of ester bonds which join the fatty acids to the glycerol, and this is formed by a condensation reaction between a fatty acid and glycerol. Also worth making a note that these bonds can be broken by lipase enzymes via a hydrolysis reaction, and that's the addition of a water molecule if you remember from previous slides. Now let's take a look at the structure of something called phospholipids. Phospholipids. So here's a triglyceride that we just talked about, and in a phospholipid, one of the fatty acid chains is replaced with a phosphate group. So here we take away one fatty acid and replace it with a phosphate group. So this is now called a phospholipid. We can simplify this. We have a simplified diagram of a phospholipid. It looks like this. We have a hydrophilic head and we have a hydrophobic tail. Hydrophilic means attracted to water, hydrophobic means repelled by water. We describe phospholipids as polar molecules. The fatty acids are saturated or unsaturated just as in our triglycerides. So here is a phospholipid where one of the fatty acid chains is saturated and the other one is unsaturated. Also worth making a note that a double bond in an unsaturated fatty acid will often cause a bend in that fatty acid, as you can see in the diagram above. The specification is keen that we are able to compare triglycerides and phospholipids, so we can do that now. Remember, compare means to show the differences and the similarities. So let's start with the similarities. The similarities are that they both contain glycerol. They both have ester bonds between the glycerol and the fatty acids. The fatty acids can be saturated or unsaturated. And they are all or both insoluble in water. That means they do not dissolve in water. In terms of the differences, triglycerides have 3 fatty acids and phospholipids have 2 fatty acids plus a phosphate group. Triglycerides are nonpolar and phospholipids are polar. So there's a summary of our triglycerides and phospholipids. Next, we're going to take a quick look at the emulsion test for lipids. The way this works is as follows. We have a sample that might contain lipids. It's crushed or blended. We then add a few drops of ethanol. So we add ethanol, we give it a shake. Next step is to add water. And if we get a milky white emulsion, this indicates lipid is present. So here we go, we've added our water and our ethanol, and there is our milky white emulsion, and that indicates the presence of lipid. So that's how we do the emulsion test for lipids. So that's a summary of triglycerides, phospholipids, and the emulsion test for lipids. Next, we need to learn about a very important topic which appears in lots of places in the spec. This is about proteins and protein structure. Amino acids bond to make proteins. Here are some simply drawn amino acids. These can bond together to make what's called polypeptides, or a polypeptide. The polypeptide can then go on to make protein. The general structure of amino acids. We need to know the structure of amino acids in more detail than simple shapes. So let's take one of these simple shapes, replace it with the general structure, and there it is. You should be able to recognize and label the general structure of amino acids. On the left-hand side, we have what's known as the amine group, and on the right-hand side, we have a carboxyl group. The letter R represents what's called a side chain, or sometimes known as the R group. The side chain varies between 20 common amino acids, so the general structure is the same for all amino acids. The R group or the side chain will vary. Here is the structure of an amino acid showing the atom arrangement rather than just the general formula, and this is helpful for us to figure out how these bond together. So we'll draw the same thing again right next to it, and we can see that these bond by a condensation reaction. That's the removal of a molecule of water. Then we have the formation of the bond. This here is called a peptide bond. And when we join 2 amino acids together, we get what's called a dipeptide. Remember, the condensation reactions happen between the carboxyl group of one amino acid and the amine group of another. Let's just add that diagram back so you have that for your notes. We need to know the different levels of structure of proteins. We start off with the primary structure of proteins. This is the specific sequence of amino acids joined by peptide bonds. This will form a chain of amino acids called a polypeptide. The secondary structure. Here is a short section of a polypeptide, and here is another short section of a polypeptide, and we can see that these can be joined by hydrogen bonds. So if we represent these polypeptides with little red stripes, we can see how they actually form secondary structures. Hydrogen bonds can be within the same polypeptide chain to form an alpha helix structure. There's our alpha helix structure, and there's our hydrogen bonds holding the alpha helix structure together. Hydrogen bonds can be between different polypeptide chains. This is to form beta pleated sheets. So here is our beta pleated sheet. Here's our hydrogen bonds holding 2 polypeptides together, and we can just add the label of the polypeptide chains there. This is the secondary structure of protein. Remember, hydrogen bonds are weak, but there are many of them. That means if we have a lot, we have stability in the structure. Tertiary structure. This is the joining of secondary structures, and this can lead to 3D tertiary structures. Here is a diagram of a It's supposed to be a 3D structure. It doesn't look too 3D, but this is supposed to be a 3D structure. And tertiary structures are held together by bonds between the R groups. There's three main types of bonds that we need to know about. The first one is hydrogen bonds that we've already mentioned. These are weak, but there are often many of them. Ionic bonds, these are strong. And we have something called disulfide bridges. These are a type of covalent bond, and they are strong. We must know and remember these three types of bond that hold proteins in their 3D tertiary shape. Examples of proteins with tertiary structure are enzymes. The next level is the quaternary structure. This is when there are two or more polypeptides joined together. An example of protein with quaternary structure are antibodies. So that's how amino acids join to make polypeptides via the formation of peptide bonds and the levels of structure of protein. Next, we're going to take a look at the test for the presence of protein in a food sample. We do the Biuret test for protein. So here we have a sample of crushed or blended food that we suspect might have protein. We add some Biuret solution, which is a blue color, and we look out for a color change. A lilac color indicates the presence of protein. So we add Biuret solution, and a lilac color indicates that protein is present. That's the Biuret test for protein. Next, we're going to take a detailed look at enzymes. These are proteins with tertiary structure. We just mentioned that. Enzymes are biological catalysts. That means they speed up chemical reactions in living things. They do this by lowering the activation energy of a reaction. They can be extracellular, in other words, work outside of cells. So here is a cell, and extracellular enzymes can be secreted by the cell. Extracellular enzymes, for example, digestive enzymes. They can also be intracellular. That means they work inside of cells. So enzymes can be released through protein synthesis, and these might be, for example, respiration enzymes, enzymes involved in respiration. Enzyme action. You may have learned at GCSE about the lock and key model. This is where the active site is a rigid 3D structure, but we have a more accurate model in terms of how they work. This is called the induced fit model. Here we have an enzyme molecule. Here is a substrate, and at first, the substrate does not fit the active site. However, the active site changes slightly to accommodate the substrate. We then get the formation of an enzyme substrate complex. This allows the product to be formed. Here's our product. So we can summarize that like this. Before the reaction, the active site and substrate are not complementary. The tertiary shape of the active site changes as the substrate binds. The tertiary structure of the active site combines with the complementary substrate. The stressing and distorting of bonds in the substrate leads to a reaction. That will get you the marks on a question about how the induced fit model works. The action of enzymes can be affected by different factors. Factors affecting enzyme action. Imagine we have a chemical reaction going on that's controlled by enzymes in a flask. So here you can see the enzymes and the substrate interacting inside the flask. The reaction conditions affect the rate of the reaction. The different factors are substrate concentration, temperature, pH, enzyme concentration, and inhibitor concentration. Let's take a look at substrate concentration first. As the substrate concentration increases, the rate of the reaction increases until a maximum is reached. Here's a graph showing the details of that reaction. Substrate concentration along the bottom and rate of reaction along the side. As the substrate concentration increases, the rate of reaction increases. This is because there are more collisions as more substrate molecules become available. Here is a low substrate concentration at the bottom end of the graph, and here is a higher substrate concentration at the further end of the graph. The rate of reaction eventually reaches a constant rate. This is because all of the active sites are occupied. So here you can see an excess of substrate, and most of the time all of the active sites are occupied. Increasing the substrate concentration has no effect on the rate of reaction. Remember, the reaction has not stopped, it is proceeding at a maximum rate. Let's take a look at enzyme concentration. Another graph, rate of reaction versus enzyme concentration. The rate of reaction increases as the enzyme concentration increases. And we can show it like this. At low enzyme concentration, we have a small number of enzyme molecules per unit volume. And at a high enzyme concentration, we have more molecules of enzyme per unit volume. So the rate of reaction increases as enzyme concentration increases. That's because there are more enzyme molecules available, so there are more frequent collisions between enzyme and substrate molecules. Enzyme-substrate complexes are formed faster. The rate increases until the substrate runs out. So on the graph, if the substrate runs out, that's what would happen. Let's take a look at temperature. The rate of reaction increases to an optimum as the temperature increases. Here's our graph, temperature versus rate of reaction, and we can see as the temperature rises, the rate of reaction rises. This is because enzyme and substrate molecules have more kinetic energy as the temperature rises. We can see low kinetic energy in the enzyme here. If we then raise the temperature, we have much more kinetic energy with the enzyme and also the substrate molecules. So enzyme and substrate will collide more frequently and form enzyme-substrate complexes more often. But at high temperatures above optimum, bonds in the enzyme are broken. So here's our enzyme molecule. High temperatures cause the bonds to break and therefore causing the active site to lose its tertiary shape. The substrate no longer fits the active site. The enzyme is denatured. The rate of reaction reduces rapidly at temperatures above the optimum. There's our graph showing a reduction in rate at temperatures above the optimum. Next, let's take a look at the effect of pH. Again, here's our graph, pH going from 0 to 14 with 7 in the middle, 7 being neutral, and the rate of reaction along the side. Enzymes have optimum pH values. Many enzymes' optimum rate is pH 7. Enzymes denature at extremes of pH. So at optimum, enzymes work most rapidly. However, at extremes of pH, either low or high, the enzymes denature. Let's have a look at the reason why. Acids contain hydrogen ions, H+ means hydrogen ions, and alkalis contain hydroxide ions, OH-. Hydroxide ions and hydrogen ions can interfere with ionic and hydrogen bonds, causing them to break. This is what causes the denaturing of the enzymes at extreme pHs. But different enzymes can have different optimum pHs. For example, we have stomach enzymes which have an optimum at a low pH, and we have small intestine enzymes which have an optimum at a higher pH than 7 in alkali conditions. So those are the factors that we need to know about that affect the rate of enzyme-controlled reactions. Next, let's take a look at inhibitors. First, we have competitive inhibitors. Here is our enzyme molecule, here is our substrate, and here is our competitive inhibitor molecule. A competitive inhibitor molecule has a shape that is similar to the substrate. Remember to say the shape is similar to the substrate, not the same as the substrate. This can fit into the active site and block the substrate from entering the active site. It competes for and binds to the active site and prevents formation of an enzyme-substrate complex. However, high substrate concentration can overcome the effect of a competitive inhibitor. So here we have two diagrams. The first one shows a low substrate concentration with the presence of inhibitor, and the second one shows high substrate concentration with the presence of our inhibitor. If we were to take a look at a graph, substrate concentration versus rate of reaction, we can see what happens when there's no competitive inhibitor present, and this is very similar to the graph we saw before. But if we add a competitive inhibitor, shown in that blue color, we can see the rate of reaction is generally lower until the substrate concentration gets high. At this point here, there's a reduced chance of the inhibitor binding to the active site because it is competing with a huge concentration of substrate molecules. Non-competitive inhibitors. Here we have an enzyme, here is our substrate molecule, and here is our non-competitive inhibitor. This fits into a site that is not the active site and stops the substrate molecule from binding. So the inhibitor attaches to the enzyme at a site other than the active site. You may have seen it called an allosteric site, but this is fine for our explanation. The tertiary shape of the active site is changed. An enzyme substrate complex can no longer form. High substrate concentration does not overcome the effect of a non-competitive inhibitor because it is not competing for the active site. So here we have low substrate concentration, and here we have high substrate concentration, but the inhibitor can still impact the enzyme even though the substrate is in high concentration because it's not competing for the active site. If we took a look at a graph, we have rate of reaction versus substrate concentration. Again, this is no non-competitive inhibitor present in black. And in red, we have with non-competitive inhibitor. Higher substrate concentration does not increase the rate very much at all. So that's an overview of what enzymes are, how they work, and factors that affect the rate of enzyme-controlled reactions. Next, let's take a look at the structure and replication of DNA. Structure of DNA. DNA is a polymer made of two strands called polynucleotides. The polynucleotides are made of individual units or monomers called nucleotides. Here is a nucleotide. We should be able to recognize and label one of these. This is a nucleotide. The pentagon shape there is called deoxyribose. This is a pentose sugar, in other words, a 5-carbon sugar. Here is the phosphate group as shown by that circle. And here we have a nitrogen-containing base, as shown by the rectangle. There are 4 nitrogen-containing bases that we need to know about: adenine, cytosine, guanine, and thymine. We can represent these by capital letters. Nucleotides can join together by condensation reactions. So here is one nucleotide, here is another one. We have the loss of or removal of a water molecule via a condensation reaction, and we have the formation of a bond. This is a phosphodiester bond. Phosphodiester bond is formed from a condensation reaction between the phosphate group of one nucleotide and the deoxyribose sugar of another. That will get you the 2 marks that you will probably see a question for if they ask you about bonding between nucleotides. Also important to remember, this reaction is catalyzed by the enzyme DNA polymerase. Let's take a look at the structure of DNA. Here we have some nucleotides. In DNA, polynucleotide strands run in opposite directions. Here's our second strand, running in the opposite direction. We call them, or we say they are anti-parallel. Bases on opposite strands are complementary. A is complementary to T, and C is complementary to G. Hydrogen bonds hold complementary bases together, so we can add those to our diagram. Hydrogen bonds are weak, but there are many of them, which gives stability to the DNA molecule. Here's our hydrogen bonds drawn in on our diagram. These are between the bases. We can simplify our DNA molecule, so there it is simplified, and we can show how it twists to make our double helix. DNA has a double helix structure, 2 polynucleotide strands held together by hydrogen bonds. The structure of DNA was discovered in 1953 by James Watson and Francis Crick, often referred to as Watson and Crick. We need to know the structure of another nucleic acid. This is RNA. The nucleotide for RNA is slightly different to that in DNA. We have a phosphate group as before. We have a nitrogen-containing base as before, but this time the sugar is ribose. This is also a pentose sugar, but it's a different sugar. The nitrogen-containing bases are also slightly different. We have adenine, cytosine, and guanine, which are the same as in DNA, but we have uracil instead of thymine. We can use letters to represent these as well. U represents uracil. So here is a diagram of the structure of our RNA molecule. It's single-stranded and it has alternating sugar and phosphate. RNA is single-stranded and is relatively short compared to DNA. And here's our simplified version of our RNA molecule. We'll learn more detail about RNA as we go through different chapters of A-level biology. Let's take a look at DNA replication. Firstly, why is it important? Well, when cells divide by mitosis for growth or repair of tissue, an accurate copy of the DNA is made for each brand new cell. Let's take a look at an overview of how this works. So we have our DNA molecule there, and this will split to produce two template strands, which are the original strands of DNA. We have two new strands that are made. We have then two new DNA molecules. The new DNA molecules are made of one original strand and one new strand. This is semi-conservative replication. Let's have a look at the details of semi-conservative replication. The steps. So here is a detailed view of our DNA. The first step involves an enzyme which will then separate the strands. So this enzyme, we do need to know about this. It's called DNA helicase, and it will unwind the DNA and, importantly, break the hydrogen bonds. You must mention the idea of breaking the hydrogen bonds to get a mark for a question about the function of DNA helicase. So step 1, DNA helicase separates a strand by breaking hydrogen bonds. Step 2, each strand acts as a template, something to copy from. Here are some free nucleotides. Next is the production of the second strand. This is controlled by a second enzyme. We need to know the name and the function of this second enzyme. This is DNA polymerase, which we mentioned earlier. This forms phosphodiester bonds between adjacent nucleotides. So, let's summarize that in our notes. Point number 3, free nucleotides are attracted and attached by complementary base pairing A to T and C to G. DNA polymerase joins adjacent nucleotides via a condensation reaction. Hydrogen bonds form between the complementary bases. Remember, the DNA polymerase joins nucleotides together and hydrogen bonds joins bases together. Just another point mentioned in the spec. Watson and Crick suggested the theory of semi-conservative replication. Two scientists, Messelson and Stahl, carried out experiments to confirm that DNA was replicated in a semi-conservative way. Next, we need to take a look at ATP. ATP stands for adenosine triphosphate, and we describe this as a nucleotide derivative. Here is a structure of a molecule of ATP. We have a rectangular or square part there, which is called adenine. We have ribose, just like we did in RNA, a pentose sugar. And we have phosphates times 3. We have 3 phosphates added on. Hydrolysis of ATP. This is the addition of water. This is a very important chemical reaction in living things. Here's our ATP. We have the action of an enzyme called ATP hydrolase. This will release an inorganic phosphate from the structure of ATP. That leaves us with ADP. So we end up from ATP with ADP and inorganic phosphate, abbreviated as PI. This releases energy for reactions in cells. So a very important reaction. Synthesis of ATP. We need to be able to make ATP as well. And this is done by a condensation reaction. So here is our ADP. Here is our inorganic phosphate. And these can join together with the help of an enzyme called ATP synthase. So those are joined together. We now have ATP. And this happens during photosynthesis or during respiration. This is how we make ATP. So a little summary of that. ATP can break down into ADP plus PI. This is a hydrolysis reaction and involves energy release. ADP plus PI into ATP. This is a condensation reaction, and this requires energy input. And that energy input comes from either photosynthesis or respiration. Hydrolysis is controlled by the enzyme ATP hydrolase, and the condensation reaction to produce ATP is controlled by the enzyme ATP synthase. Apart from producing new ATP molecules, that inorganic phosphate can also be used to phosphorylate molecules to make them more reactive. And we'll see how that works in later topics. So that's an overview of the structure of DNA and the replication of DNA, and the structure, function, and production and breakdown of ATP. Next, we're going to take a look at the importance of water. Water. This is a major component of cells. Most of the mass of the cells is made of water, and we need to explain 5 different ways in which water is important for sustaining life. Water is a metabolite. It is produced in condensation reactions, and it is used in hydrolysis reactions to break molecules apart. Secondly, water is a solvent that allows, 1, metabolic reactions to occur in living things, in cells. So here is a cell. If we were to look into the cytoplasm, we might see some molecules interacting. So here we have some molecules that interact in cells. And the particles in solution are able to move. Because they are dissolved in water, they are able to move and collide and react. This might be, for example, enzymes and substrates. Water is a solvent that also allows substances to be transported. That's in both plants and animals. Examples of substances transported by water are glucose and amino acids that would be in solution. Particles in solution can be transported through the organism. Next, water has a high heat capacity. This means water absorbs or releases a large amount of heat energy before its temperature changes. That means water is a buffer to temperature change. This means it resists rapid increase or decrease in temperature. So if the temperature of an environment was to change, for example between day and night, there's a big change in temperature often between day and night, and high heat capacity means that there is a resistance to rapid decrease or increase in temperature. 4. Water has a high latent heat of vaporization. This means water absorbs a lot of heat energy before evaporating. When it evaporates, it carries that heat energy away. So, an example would be through sweat. Here we have water evaporating from the surface of the skin, and as it does that, it carries heat away from the body. And because it's absorbed a lot of heat energy in order to evaporate, that heat energy will be carried away into the environment. It has a large cooling effect for a small volume of water loss. Next, water has strong cohesion between its molecules. So the example we're going to look at is xylem, which transports water up plants. So here is a water molecule, and one important feature of water molecules is that they have a positive end and a negative end. We describe these as delta negative and delta positive. This is because they are positive and negative relative to each other. Water molecules have hydrogen bonds between them, which gives them cohesion. They stick together. So this cohesion allows for the formation of hydrogen bonds. There's our hydrogen bonds in our diagram. Cohesion allows long, unbroken columns of water to form in plants. Water can reach the top of even the tallest trees via a transpiration stream, and the water columns remain unbroken because water molecules stick to each other by cohesion. Cohesion also produces surface tension on the surface of water. Here is some water. You can see the surface over here, and the water molecules are held together by cohesion forces that forms hydrogen bonds to provide surface tension. Surface tension supports small animals on water. So here is what's called a pond skater, and it's supported on the surface of water by tension in the surface of the water. So that's an overview of the whole unit of biological molecules for AQA A-level biology. If you found this video useful, please do subscribe and like. That will encourage the production of more videos. But other than that, thanks for watching, and I'll talk to you again soon.